Good morning, students. Welcome to B.Sc-II 4th semester Zoology class. In the last class, we were discussing Meiosis I. So today we shall discuss the Meiosis II. Meiosis II. So Meiosis II starts from interkinesis. Interkinesis is similar to that of interphase, but during interkinesis, what happens, the duplication of chromosome doesn't occur. Whereas in interphase, duplication of chromosome does occur. So multiplication of the DNA takes place in interphase, but during interkinesis, there is no multiplication of DNA or chromosome. Next, Meiosis II. Again, Meiosis II is subdivided into prophase II, metaphase II, anaphase II, and telophase II. So prophase II is different from prophase I. We already discussed the prophase I. Prophase I is further subdivided into 5 stages such as liptotin, zygotine, pachytine, diplotin, diakinesis. But here the prophase II is similar to that of mitosis. The Meiosis II is known as equational division. Meiosis I is reductional division. So at the end of Meiosis I, chromosomal number is reduced to half. The number of chromosomes that are present in their parental cell is reduced to half in their daughter cells in Meiosis I. Therefore, the Meiosis I is called as reductional division. Whereas in Meiosis II, reduction doesn't occur, equational division takes place. Equational division. So the number of chromosomes that are present in the parental cell will be remaining in their daughter cells. So that is the beauty of Meiosis II. So prophase II. Meiosis II is the second part of the meiotic process. The Meiosis II consists prophase II. Each dyad is composed of a pair of sister chromatids attached by a common centromere. Metaphase II. So each dyad in prophase, each dyad is composed of a pair of sister chromatids attached by a common centromere. So each chromosome has 2 sister chromatids. Each chromosome has 2 sister chromatids. Okay, and both are attached at the common centromere. Then metaphase II. Metaphase II. Centromere are positioned at the equatorial plane. Centromeres brought on the equatorial plane in metaphase II. Anaphase II, centromere divides and the sister chromatids of each dyad are pulled to opposite poles. Here, look here. So this is prophase II, metaphase II. So all chromosomes are aligned on equatorial plane. So each chromosome has 2 chromatids, and both chromatids are attached at the center where the centromere is present. So anaphase II, centromere is split and chromatids, the sister chromatids are separated. They are moving towards opposite poles. Okay. Anaphase, centromere divide and the sister chromatids of each dyad are pulled to opposite poles. Telophase II, one member of each pair of homologous chromosome present in each pole. Okay, each chromosome is referred to as monad. Now, each chromosome is referred to as monad because it is having only one chromatid. Dyad become monad. A combination of maternal and paternal genetic information. So each monad is a combination of paternal as well as maternal genes. So nuclei reform around chromosomes at the poles. Nuclei formed here. Nuclei formed. Following cytokinesis, the finally 4 haploid gametes result from the single meiotic event. So from each cell, 2 cells are formed. 2 daughter cells are formed. Can you see here? So totally 4 gametes are formed. 4 daughter cells are formed at the end. Okay. So around each group of chromosome, nuclear envelope is formed. Nucleolus is formed inside the nucleus, and the haploid cells are formed at the end of meiosis II. Meiosis II. So functions of meiosis: production of haploid gametes so that fertilization restores the normal somatic chromosome number. So during meiosis, what happens, diploid number of chromosome is reduced to haploid, right? Haploid. When haploid gametes are fused during fertilization, the normal number, diploid number of chromosome is restored in the somatic cell. Again it becomes 2n number. So our body cell has 46 chromosomes, but our gametes have 23 chromosomes. 23 from sperm, 23 from egg. Both together it becomes 46. Therefore, the formation of haploid gametes is necessary for maintaining normal somatic chromosome number in all the individuals of a species, and that is continued from generation to generation. Production of tremendous amounts of genetic variation. So genetic variation occur during meiosis because during packaging stage crossing over takes place. As a result, the paternal as well as maternal genes are aligned on the same chromatid. Segregation of the 2 alleles of each gene. This takes place due to pairing between the 2 homologs of each chromosome and their separation at the first anaphase. Recombination between linked genes due to crossing over during packaging stage. Meiosis facilitates segregation and independent assortment of chromosomes and genes. So meiosis facilitates segregation, separation, and independent assortment. Independently, the genes are assorted, separated. They are not depending on other genes. They get separated and they aggregate to form chromosome in the next progeny. In sexually reproduced species, meiosis is essential for the continuity of generation because meiosis results in the formation of male and female gametes, and union of such gametes leads to the development of zygotes and thereby new individual. So for the formation of gametes, meiosis is required, right? After the fusion of gametes, zygote is formed. Zygote undergoes cleavage to produce multicellular new individual. Now let us see the differences between mitosis and meiosis. Mitosis occurs in somatic cells, whereas meiosis occurs in reproductive cells. Mitosis, one cell produces 2 daughter cells, whereas here one cell produces 4 daughter cells. It is an equational division separating sister chromatids in mitosis, whereas in meiosis it is a reduction division. The first stage is a reduction division which separates homologous chromosomes at first anaphase. Sister chromatids separate in an equational division at second anaphase. So here only one division per cycle, that is one cytoplasmic division per equational division takes place. Here 2 divisions per cycle, 2 cytoplasmic divisions, 1 reduction division and 1 equational division. So chromosomes fail to synapse, no chiasmata formation occurs in mitosis. Whereas in meiosis, chromosomes synapse and form chiasmata. Homologous chromosomes synapse, the pairing takes place. Pairing of homologous chromosomes is called synapse. Synapses occurs and formation of chiasmata takes place. Chiasmata means point of attachment between non-sister chromatids. So chiasmata are also seen in meiosis. These two, synapse and chiasmata formation, is not seen in mitosis. Genetic exchange between homologous chromosomes doesn't occur here in mitosis, whereas in meiosis, genetic exchange through chiasmata occurs between homologous chromosomes. Then the genetic contents of daughter cells are identical in mitosis, whereas in meiosis, the genetic contents of daughter cells are different because of recombination. Chromosome number of daughter cells is the same as that of mother cell, whereas here chromosome number of daughter cells is half of that of mother cells. Daughter cells are capable of undergoing additional mitotic divisions in mitosis, whereas here the daughter cells are not capable of undergoing another meiotic division, although they may undergo meiotic division. So they have already undergone the meiotic division. Yes, first mitotic division is completed. Later on they have entered into meiotic division. Start at the zygote stage and continues through the life of the organism. Mitotic cell division starts at the zygote, means zygote undergo division by mitotic division. This is called cleavage. Okay, so because of the cleavage, zygote becomes multicellular. And here the meiosis occurs only after puberty in higher organisms, but occurs in zygote of algae and fungi. So these are the differences. Gametogenesis, yes, formation of gametes. Formation of gametes occur only through meiosis. Gametogenesis is the process of production of haploid sex cells. Gametes have one half of the genetic material, haploid chromosome numbers, from the germ cell of each parent. Fusion of spermatozoa and ova at time of fertilization results in a zygote with a diploid genome. The process of production of spermatozoa is called spermatogenesis, and the process of production of ovum is termed oogenesis. So here you can see this is spermatogenesis and this is oogenesis. Spermatogenesis, can you see here? Spermatogonium grows in size. It grows in size. It becomes first spermatocyte, means primary spermatocyte. Primary spermatocyte undergoes meiosis I to produce two daughter cells called secondary spermatocyte. Secondary spermatocytes undergo second meiotic division to produce spermatids, 4 spermatids, and this up to the formation of spermatid, the process is called as spermatogenesis. Spermatids, circular or the spherical spermatids, undergo differentiation to become tadpole shaped spermatozoa, and that differentiation is called as spermiogenesis. Okay, this is how in case of males, with the help of meiosis, from body cells, germ cells, the male gametes are produced, spermatozoa are produced. All spermatozoa are functional. All 4 spermatozoa are functional. So one cell in meiosis, spermatogenesis produces 4 sperms, functional sperms. Whereas in oogenesis, what happens here? Oogenesis, oogonium becomes larger in size during growth period, and this is called primary oocyte. Primary oocyte undergoes first meiotic division to produce 2 unequal sized cells. One is larger, another is smaller. Smaller one is called the polar body, first polar body. Larger one is called secondary oocyte, and secondary oocyte undergoes second meiotic division to produce one oocyte and second polar body. So by that time, first polar body also undergoes second meiotic division to produce 2 polar bodies. Totally 3 polar bodies are formed, one oocyte, and this is ovum proper. So at the end of oogenesis, from one cell, only one functional ovum is produced. 3 polar bodies are produced, and these polar bodies are degenerated later. So this is about the gametogenesis. Now let us see one video clipping is there through which we can understand the all stages of meiosis II, how the 4 haploid gametes are produced at the end of meiosis II. Let us see it.
Meiosis II. In this module, you will learn about Meiosis II. Have you ever wondered how the chromosome number in humans is conserved generation after generation? Well, this is achieved by a type of cell division called meiosis. Meiosis is a specialized nuclear division in which the chromosome complement of a cell is halved. Meiosis takes place inside the gonads of sexually reproduced cells, all of which are haploid. Meiosis involves 2 steps, meiosis I and meiosis II. Meiosis I is the reductional division in which the chromosome number gets halved. Meiosis I is followed by meiosis II after a very short interphase called interkinesis, during which DNA replication does not occur. Unlike meiosis I, meiosis II is an equational division. In this module, you will focus on meiosis II. Each daughter cell formed after meiosis I inherits only one of the 2 homologous chromosomes, and each homologue has 2 sister chromatids. Meiosis II involves the separation of sister chromatids to produce haploid cells. Therefore, meiosis II is an equational cell division. As both the daughter cells produced from meiosis I undergo meiosis II, the final result of meiosis II is 4 haploid daughter cells. Like meiosis I, meiosis II can also be divided into 4 stages. These are prophase II, metaphase II, anaphase II, and telophase II. Prophase II is very short. During this phase, the nuclear envelope and the nucleoli disappear, and the spindle is formed with the help of the microtubules. In addition, the chromosomes recondense during the prophase II and become compact. Prophase II is followed by metaphase II, during which the chromosomes align along the equatorial plate and the microtubules attach to the kinetochores of the chromatids. Metaphase II is followed by anaphase II. In anaphase II, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubules become shorter. Anaphase II is followed by telophase II, which also marks the end of meiosis II. During telophase II, the nucleoli are reformed and the haploid nuclei get enveloped in membranes. In addition, the chromosomes decondense and the spindle disappears. Division of the cytoplasm Cytokinesis results in 4 haploid daughter cells. Meiosis plays a significant role in the life of sexually reproducing organisms. It results in gamete formation and helps conserve the chromosome number in sexually reproducing organisms. All the 4 gametes produced are genetically unique as they are produced by the crossing over of homologous chromosomes. The genetic uniqueness of the gametes is further attained by the independent assortment that results in each daughter cell receiving either homologue from each chromosome pair. Let's summarize what you have learned. Meiosis is a specialized nuclear division in which the chromosome complement of a cell is halved. Meiosis involves 2 steps, Meiosis I and Meiosis II. Meiosis II has 4 stages, prophase II, metaphase II, anaphase II, and telophase II. Prophase II is very short. The nuclear envelope and the nucleoli disappear and the spindle is formed. The chromosomes recondense and are compact. During metaphase II, the chromosomes align along the equatorial plate and the microtubules attach to the kinetochores of the chromatids. In anaphase II, the sister chromatids separate out and move towards the opposite poles. During telophase II, the haploid nuclei gets enveloped in membranes. The chromosomes decondense and the spindle disappears. Cytokinesis results in 4 haploid daughter cells.
Okay. So assignments. First assignment is explain how the interkinesis is different from the interphase in meiosis. Second one, write the differences between prophase 1 and prophase 2. Okay. So I hope you have followed the entire meiosis cell division. Thank you.